get started now. Thank you all for coming, and I'm going to turn it over to Demetri after just a couple slides real quick. I'm with MUSC Sustainability. We put on these events once a month. Just a little bit about what we do. We do recycling and waste management here at MUSC, energy and water conservation, food, transportation, climate, and green building. And then if you want to hear more about events like this, we have a green team sign up at the front. That's our monthly newsletter. We're also on social media at MUSC Go Green, and you can find us on Yammer at broadcast-sustainability. And with that, I'm going to turn it over to Dr. Spyropoulos. Thank you.
All right. Well, thanks for coming, showing up. I'll try to be brief to leave time for questions. So the title of my talk is Obesogen Toxicology: What Doesn't Kill You Could Make You Fatter. Okay, and currently there are very few safety nets in place for what we're exposed to in the pharmacy and grocery store. So if you picture a time before seat belts and airbags, that's to a great extent, that's where we still are when you go to buy food products. And this is based on old toxicology, which is lethal dose. Lethal dose in which half of the animals die during exposure, or lethal dose in which half the cells in culture die. And so I saw this cartoon, Ziggy cartoon, about 2 weeks ago, and I said, wow, that's not funny at all. That's actually really, really true, because there are many products that are generally regarded as safe based on this. And so you have, for example, homogenized milk. You have listed as ingredients milk, vitamin D3, but what I'll tell you about is this compound which we think is an obesogen, which is not listed on the label, but is in the definition of homogenized. So these products have no observed adverse effect in animals and in cells at certain doses, and they're therefore considered safe. In modern toxicology, we go beyond lethal dose and look at long-term health effects. Okay. Okay. So currently, whoops. Yeah. Currently, we have roughly half of Americans are diabetic or borderline diabetic, and in 10 years, half of Americans will be obese or morbidly obese. And there is an interesting parallel between the bulk and variety of synthetic chemicals that are produced and the incidence of diabetes and obesity. And you can see in 1990, this is the rate of obesity. In 2010, it's increased dramatically. And now, currently, approximately 34% of Americans are diagnosed as obese. And so we, in the last 4 billion years, we've evolved based on our genetics, our niche, and our exposure to the environment. And many of these synthetic chemicals we've never come across before. Okay, so what are some fundamentals? So on a basic level, it's very simple. The amount of calories that you take in versus the amount of calories that you burn is going to determine whether you gain weight or lose weight. So obesogens drive a variety of aspects of a positive caloric checkbook, okay? And so they're chemicals that increase your hunger or decrease your satiety. They can slow metabolism. They can alter nutrition, alter the gut microbiome, and also disrupt stem cell differentiation. So in the fetus you have, and the adults, you have mesenchymal stem cells, and those can differentiate, for example, multiple directions, but two of the directions they can differentiate into are osteoblasts or adipocytes. And with our studies, we see a shift from bone formation to adipose tissue formation. Okay, but when you go to the human population, you're going to see that different people are exposed to different levels of obesogen, different types of obesogens, and different combinations. And there's also a genetic susceptibility, which may include deficiencies in certain compounds. Now, if you see a family with a history of obesity or diabetes, that does not preclude genetics alone. So genetic susceptibility may be part of it, a major part of it, but it could also be where those individuals live and what their exposures are, lifestyle. Okay. All right, so our lab, I was trained as a developmental biologist, and one feature that I've been studying is body pattern development and the patterning of the organism. And we focused on Hox genes, which are conserved throughout metazoa. So for example, here are Hox gene activities within a fruit fly, and that anterior, posterior, proximal, distal, dorsal, ventral patterning is conserved up through humans. But what my first graduate, so we were knocking out these genes, and what my first graduate student found out was that shifting ovarian hormone levels actually changed these fundamental patterning gene expression patterns. And what you see here on the right is an experiment we did in which we exposed to pregnant female mice to a similar relative dose as pregnant women to a chemical called Makina, which is 17-hydroxyprogesterone caproate. And that is a chemical given to women who are at risk for preterm birth. And what we did, what you see here, are gene expression arrays in the mammary glands of the offspring female from mothers who are treated with Makina or not treated with Makina. So what you can see from the back of the room is the gene expression profiles within the mammary glands of the offspring females is very different based on that exposure. Of course, preterm birth is a very horrible thing, so it's a give and take phenomenon. So more recently, I met Lou and Buzzy Gillette, and they got me more excited about environmental endocrine disruptors and the idea that there are a variety of systems within the body that are sensitive to these disrupting compounds. And the nervous system is arguably one of the most sensitive or easily measured phenotypic shifts. The immune system, metabolism, including obesity, diabetes, and metabolic syndrome, and reproduction. So these are major areas of research having to do with environmental hormonal disrupting chemicals. And I just wanted to show, Buzzy did a study of families from Yaqui Valley in Mexico, and this was one of these mega farming industrial communities, and there were people who lived in the valleys and people who lived in the foothills. Those in the valleys were exposed to pesticides, herbicides, and other environmental chemicals, whereas those in the foothills lived in what were considered more original environments. And so I like this example because it's a very simple, elegant questionnaire. Basically, the cost for doing this study was the cost of a pencil and paper, and then collecting these children and having them draw pictures of the family. And on the left side, you'll see pictures drawn by 4- and 5-year-olds family members, and on the right-hand side, you'll see kids who were from the valley, and you can see a dramatic difference. And so this pointed out that there are significant neurological disorders in the children growing in the valley, and later studies showed that cord blood and breast milk had multiple pesticides in them. So it prompted much more research and better regulations. Okay, so with Lou, he said, well, why don't you take your preliminary data and we'll take my preliminary data and we'll apply for this Gulf oil spill project and say that we're gonna ask the question, are there obesogens released from the oil and dispersant? So the Gulf oil spill involved 200 million gallons of crude oil and 2 million gallons of Corexit dispersants that were used to emulsify and break up the oil so that it wouldn't form oil slicks on the coast. And one idea was that if the dispersant broke the oil up into smaller droplets, then it would be more bioavailable to oil-eating bacteria. Of course, it turns out Corexit is toxic to those bacteria. So it would have been good to know before we put 2 million gallons. This is a combination of dispersants, of emulsifiers, and organic compounds. And actually, it's because of its effect on your lungs. If I had a tablespoon and I spread it over the theater here, we'd have to evacuate because people would be coughing and breathing poorly. So what we did was we went about determining whether there were obesogens present and what those were in that complex mixture, crude oil, Corexit, and the emulsified mix. And what we did was we took advantage of a feature of the basic biology of obesogens, is that they can often function as ligands for these transcription factors, PPAR and RXR. So we set up a reporter assay in which if a ligand bound to the PPAR ligand binding domain, we would transactivate and get luciferase luminescence. And this is quantifiable. So what we can do is fractionate this mix and see where the activity lies and follow it through to identify the compound. And we actually found a couple of compounds, and I'm going to focus most of the talk on this one, which is DAS, dialkyl sodium sulfosuccinate. So it acts as a PPAR ligand, and it binds to the binding pocket, and it transactivates. This is an amphiphilic compound. So it has hydrophobic domains and hydrophilic domains. And so it in itself will form a lipid bilayer, but will also embed in lipid bilayers and is a very good emulsifier. Okay, and that was a diagnostic tool. I know this is a really busy slide, but I'll just touch on a couple of features. We also came in with a functional assay. So I mentioned how we can take mesenchymal stem cells, which have multipotential for producing, say, bone or fat, and we expose them to DAS and this other compound that we found, SPAN80, in varying doses, and saw that with increasing doses that we had increasing differentiation of human mesenchymal stem cells and mouse mesenchymal stem cells into adipocytes. And we can measure triglyceride levels with oil red O or with a fluorescent tag. So we can quantitate that as well. And we see a shift in gene expression saying that they're losing their stemness and going down that differentiative pathway. So DAS is not only working within that transactivation assay, but is also acting functionally in vitro in these cells. Okay, so what is DOS found in? Well, as I mentioned before, it's not necessarily listed on labels. It's an additive, it's an emulsifier wetting agent for insoluble compounds. So in drinks, it's a coloring agent, flavoring agent, oils, fats. Those all need to be emulsified into that liquid. Fruit drinks, sodas, salad dressings. If you take an oil and vinegar salad dressing and you shake it up and you put it down and you come back half an hour later when you're finished shopping and it's still mixed up, it may be in there as an emulsifier. Okay. The cosmetic industry a couple years ago repealed a ruling that they had on limiting the amount of DOS that could be in personal care products, and now you have eyebrow pencils that are 7 to 10% DOS. And I remember when I was a kid, my mom used to lick the eyebrow pencil before she would apply it. I guess you don't have to lick it anymore. So, okay, but these were based on studies in which a 0.4% solution was used, and what they found was there was no reduction in the live birth numbers. And that's what all of these studies are based on. So I'm going to go into studies where we go with less than 1/100th that dose and the effects on the offspring. Okay. Cows are also fed DOS to remove ciliated protozoa and change their biome. And, you know, so the question is, what else are they changing in the gut biome of the cows? Well, they grow faster, they fatten up, they produce richer milk. But of particular interest, when my graduate student presented to OBGYN, they said, oh, well, this is cholase or docusate stool softener, which is taken by 35% of pregnant women in doses of 50 to 500 milligrams a day to alleviate symptoms of constipation, which are very common in pregnancy. There tend to be higher rates of use and dosages among non-Hispanic blacks, and they're also twice as likely to be anemic, and the iron supplements will drive constipation, and so they add DOS in there to curtail an increase in constipation. So this is really, it's scary, but it's also nice for our fetal origins model, which is that the fetus or the young child is more susceptible because they carry many more stem cells for epigenetic modification. Okay, so our animal models, so we go from in vitro cell culture to now animal models, and we use this inbred C57 Black 6 as our workhorse, but because humans take it, we can use humans in the study as well. But more recently, because we're interested in susceptibilities, we're going with outbred strains of mice that have sensitivity and resistance to metabolic disorders and also neurological disorders. So we have a variety of phenotypes that we look for, and our biological questions are genetic susceptible populations. We can do that with Peromyscus in humans. Mechanisms we can develop in all three. Okay, and just a point, because my summer student is here, these chemicals behave differently, not only based on genetic susceptibility in human populations, but within organisms. And these endocrine disrupting compounds will drive imposex in snails. So now with the dredging of the harbor for the super tankers, we're going to be bringing up these legacy disruptors like tributyl-10, and so we can monitor water quality by the rate of imposex in the snails. Okay, so here's the the animal experiment. And basically, we wanted to mimic human exposure. So women start taking Colace or Docusate mid-pregnancy, and they may take it through during nursing as well. And so we went with that dosage, which was about 1/100th the amount, which is more on par with what women take when they take that. But then after these mice are weaned, we feed them a normal diet, ad lib, so they can eat as much as they want, and just water. And then we follow adiposity, lean mass, glycemic index at 12 weeks of adulthood and at 16 weeks. So these are persistent changes that we're looking for. Okay, and I know this is a complex slide, but by looking, by measuring fat grams, bone area, and lean grams, we see no change in lean mass. We see a reduction in bone area and an increase in fat grams. So that's consistent with our stem cell model of differentiation. So the offspring of DAS moms have increased adiposity, body mass, and decreased bone area. If you do glucose tolerance tests, they're glucose intolerant. And also if you measure insulin response, under fasting conditions, they have higher resting insulin, and they're less responsive to a dosage of glucose. Okay, and then just to continue on, offspring are hungrier, they have higher levels of leptin, and they're metabolically obese and have chronic inflammation from circulating inflammatory cytokines and adipogens. And finally, probably what I consider the most exciting results is that if you look at circulating lipid profiles, you see profiles that are very similar to adult mice fed long-term with a high-fat diet. So even though these are being fed a normal diet, their circulating lipid profiles are the same as if a mouse were on a high-fat diet for 14 or 16 weeks. Okay. And so mechanisms. Well, I talked about the stem cell differentiation. Okay, and I'll finish up real quick. So we're looking at target organs: liver, gallbladder, GI tract, mammary gland, and target systems: metabolism, nutrition, and microbiome. So patients orally dosed have circulating levels within 3 to 4 hours, which are well above the levels that we expose cells to. And let's see. And then we see liver energetics. These are human hepatocytes. And so their energetic potential and oxidative stress are increased within these. Okay, so, and we see alterations in breast milk nutrition, milk fat globules. Okay, so with that, I'll, the one point that Ari Shapiro said he was considering interviewing me for this, and one thing that I didn't have when I gave the talk to him was personal empowerment. The idea that before the FDA makes changes, well, as Linda Birnbaum said, you can't change your genetics, but you can change your environment. So you can navigate through the system and perhaps decrease your exposure to obesogenic compounds and improve your nutrition so that you reduce these effects. Okay, so the people, most of this work was done by Lexi Temkin, a PhD student that just got her degree, a postdoc Rob Bowers, myself, John Botts, and Danny Newton. And I don't have a picture of Danny Newton there. So, so with that, ta-da. Okay, so, and our funding source was the Gulf Oil Spill Project. Okay, how far over did I go? Oh, whoops. Okay, so I guess we'll take questions at the end because,
Good idea. So thank you for attending this afternoon. My name is Mark Hammond. I'm with Drug Discovery and Public Health, and I'm actually a natural products chemist, and wanted to just talk to you a little bit today about the impact that climate change will have on, or the potential impact that it will have on human health. Let me see if I can find a pointer. So the two areas that I'd like to just cover are the sea level rise induced loss of species diversity. This is actually very important for natural product studies, because actually species diversity is a very important aspect of what we study, and the utility of natural products in the control of cancer and emerging infectious diseases. And then a second area is the increased exposure to microalgal blooms and the algal or the harmful toxins that are produced by those types of organisms. And so algal blooms have actually increased in the last decades. The carbon dioxide levels in the environment is a factor that determines how frequent blooms occur, and they have increased it to approximately 10 times what they were before the pre-industrialized area. So this slide is from literature. This just shows that there have been 5 mass extinction events in the past due to various issues. The earliest one, which is interesting, is due to the evolution of cyanobacteria, and cyanobacteria actually enriched the atmosphere in oxygen. It was fairly low here, and then enriched the oxygen levels, which caused or triggered a mass extinction event, because prior to that, the environment was primarily anaerobic. And so we are now in the middle of what scientists believe is the 6th mass extinction event. We have lost, or over a period of 300 years, if extinction rates continue to occur at the existing rate, we will essentially lose 75% of all mammal species over a period of just 300 years. And the reason this is important, or one of the reasons this is important, is because species diversity actually is an important factor in chemical diversity. This is the structure of the molecule Taxol. Taxol was discovered several decades ago from the Pacific yew tree, and this is an increasingly endangered species along the Pacific Northwest. And this molecule is arguably the most important control for cancer that we have. It's the most frequently prescribed drug for the control of cancer. And so it's a good example of unique chemical diversity that would not be available without protecting species diversity, and a very significant impact that it has on human health. Of course, there are still many types of cancer that we need to work on identifying therapeutics for. And so we continue to look at plant and animal diversity as a resource for these for potential therapeutics. And then, of course, molecules from the environment as well as synthetics are used in combinations. There are approximately 100 different drug combinations that are used in cancer chemotherapy, and plants and bacteria play an important role in this. Etoposide is from America podophylotoxin, or podophyllum, and vincristin is from Catharanthus, and this just shows the incredible structural complexity of the molecules that come from the environment, and the fact that these are resources that would not be available without any other, or from any other synthetic or commercial library. So just a couple of projects that we're looking at in our lab. This is a project that Dr. Yvette Pina is working on. This is an endangered plant from the southeastern United States. It's a golden barrel cactus. This was used by the Native Americans to control inflammation. An interesting aspect that impacts or why this is endangered is due to poaching. You don't usually consider poaching as a typical threat to plants, but because this is actually a very attractive ornamental, it's actually removed from the environment and sold. And so we've taken the approach of looking at the microbiome associated with the plant. So every single species actually is a host to a unique set of microorganisms that are very important for the production of secondary metabolites. This is a tool called molecular networking, which is an interesting chemoinformatic tool for assessing the compound diversity in a sample. And then from using the combination of this chemoinformatic tool and looking at the microbiome, we were able to successfully identify a series of alkaloids that have interesting biological activity or potential for the control of cancer. This is a sample or species which is indigenous to this area. This is an endangered plant called Obesogen canby. It's actually endangered at risk for extinction due to habitat loss primarily. South Carolina actually has the largest population of this particular species, and there are 50 populations in the state of South Carolina. And again, this is a network analysis of 46 microorganisms that live in association with the plant. And you can see these red clusters are actually unique classes of molecules that we're investigating as for their potential biological utility for the control of cancer or infectious diseases. This actually grows in lowland areas, and so it's a potential risk for sea level rise. And this slide just shows the impact of carbon dioxide as far as a energy absorbing molecule in the environment. All of you are familiar with ozone. Ozone is an important absorber of UV light in the outer atmosphere. And then, of course, carbon dioxide, I think this illustrates as absorbs a significant amount of near and thermal IR energy. And then with this combined with water, makes up some of the largest mixture of greenhouse gases in the environment. And so historically, there's been a very clear link between levels of carbon dioxide and global temperature. Let me just use the. Okay, so in the past, during the actually the first two ice ages, it's been hypothesized that most of Earth was covered with ice. This first ice age was induced by the growth of cyanobacteria and the emission of carbon or oxygen into the environment and the absorption or removal of methane and carbon dioxide. So if you compare Earth with our sister planet Venus, Venus actually is just slightly closer to the sun than Earth. It's slightly smaller in diameter, but actually the surface, which is 95% carbon dioxide, is actually extremely high pressure, 92 atmospheres, and over 800 degrees Fahrenheit. So it represents the planet in the solar system where greenhouse gases have effectively become completely out of control. So one of the factors that impacts the greenhouse gases on Earth is that as temperatures increase, so do emissions of methane from the environment. And this is particularly noticeable in the polar region. This is actually changes in methane levels that were detectable just within the last decade, and you can see that there's been a significant increase in methane emissions from the polar region or the North Pole polar region of the world. And some of the unique anomalies that have occurred as part of this are these events where there is a large emission of gas and it forms a huge crater in the Earth where from the expansion of methane gas and the emission from the environment. And so this is one of the greatest impacts, especially for an environment like ours, is sea level rise. And so this is a picture from Antarctica. I spent several seasons in Antarctica in the early 1990s, and we never witnessed, or I never saw any of these types of large waterfalls of melting ice flowing into the ocean. And So the impact, the projected impact on sea level rise, I'm sure you probably realize, but in the last 50 years or so, there's been a 5 to 8 inch increase in sea level rise in the environment, and by 2050, an additional 6 to 24 inches of sea level rise may occur. And so, in response to the additional carbon dioxide in the environment, there's an increase in actually cyanobacteria blooms. This is a cyanobacteria bloom that occurs in up near Alaska. And actually, these blooms are very valuable in sequestering carbon and excreting carbon dioxide into or excreting oxygen back into the environment. As much as 50% of the carbon from one of these blooms will be sequestered back into the sediment or into the environment, and then form essentially the coal and oil deposits of the future. So the challenge is, of course, is that these algal blooms also produce a small number of very potent toxins. This is microcystin, which is a tumor promoter, which promotes liver fibrosis and liver cancer. Just a year ago or a year and a half ago, there was a very large bloom in Lake Erie on which contaminated the water with microcystin, and the removal of these molecules is actually very difficult and rather expensive. So just recently, you may have seen this in the news, but just a few weeks ago, there was already a microcystin bloom in this reservoir in Salem, Oregon, which made the drinking water actually undrinkable or unusable to humans, with a level of microcystin, which is approximately 10 times the level which is considered safe for consumption. So I just wanted to mention some of the promising technologies. You've probably seen some of these, but actually NASA actually has a solar cell which is now 40 to 50% efficient, and actually they're trying to license this technology. A group at MIT has started using photonic crystals, which actually convert light or heat. Light into heat first, and then into light at a specific wavelength, which can be then be converted into electricity very efficiently. There's also improvements in biocrop efficiency. It turns out that one of the interesting opportunities is to use the microbiome of plants to improve efficiency, which is an area that the USDA has been very interested in. And then, of course, there's the implementation of wind and tidal electrical energy, increased use of electric vehicles, cars, trucks, and tractors. So this is my last slide. A lot of times I will hear that people will be concerned about the cost of moving to renewable energies. I'd just like to remind you that there have been years where the United States has actually spent $500 billion on foreign oil, and as much as $100 billion a year. Or Texas generates approximately $100 billion a year in energy income. So I think one of my, I feel that one of the interesting solutions is the institution of carbon credits. This is not actually a tax; it just increases the cost of carbon-based energies, and then reduces the cost of other forms of energy, which include tidal, wind, solar. And one of the interesting benefactors for a carbon credit would be the oyster industry. Oysters actually effectively sequester 40% of their weight in carbon, permanently removing that from the environment. And that's all I have. Thank you.
John Vena, I'm Chair of Public Health here at MUSC, and I'm going to tell you about a project that we're, oops, what happened here? We're not at the beginning. Here we are. So Cultivating a Healthier Tomorrow for Children in South Carolina. This is my email if you have any questions or want to talk further about this. So our team here at MUSC was just awarded a grant 2 years ago. We're just entering our third year. We just put in a renewal for 5 more years. It's called Environmental Influences on Child Health Outcomes. It's a 7-year initiative by NIH to look at early environmental exposures on child health and development. So Demetri's talk was a really great introduction to what we're interested in. And the whole idea of the ECHO program, which is out of the director's office at NIH, is to expand existing cohort studies of mothers and children. And the idea is to enroll 50,000 children across the United States into the studies and follow them over time. The 4 focus areas are upper and lower airway disease. Obesity is one, and metabolic disorders related to these endocrine disruptors, and pre, peri, and postnatal outcomes, how exposures during pregnancy impact pregnancy outcomes, both during pregnancy and just after pregnancy, and neurodevelopment. So how are some of these endocrine disruptors related to developmental outcomes in kids, including ADHD, risk for autism, et cetera. So the ECHO mission and vision is to enhance the health of a nation's children for generations to come and to be a preeminent research program in child health, and conduct observational studies and institute best practices for team science. So that's ECHO overall, and as you can see here, this is and I will post these slides. Is that possible? And you can go to the NIH or just Google ECHO, and you can go there and it'll give you an overview of the program. So it has 7 components. There's a coordinating center at Duke. There's a data analysis center at Johns Hopkins. This is an exposure core, and then ProCore is an outcomes core. So they assist the teams. There's cohort sites, Idea States Network, and a genetics core, which is to be funded this coming year. So we're part of the both the cohort sites and Idea States. I'm going to talk to you about our cohort site here at MUSC. And actually, Andy Atz is PI for South Carolina for one of 17 Idea States Clinical Trials Network. I'm not going to talk about that, but it's to translate everything we're learning in the ECHO project to clinical trials or interventions that could be done in kids to help keep them healthier. So it's really fantastic. There's those 2 components to the ECHO overall. So how is ECHO? I'm the contact PI. The other MPI is Roger Wapner, who's a fetal medicine doctor at Columbia. And the MUSC team here, and from Public Health Sciences, OBGYN, Pediatrics, and the Biomedical Informatics Center. So we have faculty, and we're collecting data on mothers and children who were previously followed to monitor the growth of a fetus, exposures, and nutritional assessments from 2009 to 2013. And so this study enrolled 2,400 children from diverse backgrounds at 10 clinical centers, including 300 here at MUSC, and Dr. Roger Newman was the principal investigator. And so when the RFA came out for these ECHO sites, this was, again, they were interested in existing cohorts. So we said, well, let's take all the children. So they wanted to study fetal growth of the mothers and the impact of how fetuses grow across 4 race sex groups. So we said, well, let's take all of these and monitor the children's health for 7 years up through early adolescence. So it was a prime opportunity, one of the best studies ever done of pregnant women and the growth of the baby during pregnancy. And so our goal was to follow the children for looking at outcomes. So it's a fantastic experiment. Our proposal was to focus on obesity and neurodevelopment, but actually we can do all of the outcomes in the study because it's a cohort study. And we'll have far-reaching effects. So we're one of 35 pediatric cohort centers that were funded. There's 84 cohorts, and so that's where they get the 50,000 kids. And so here's a link on to give you some of the news. So the goal is to determine if persistent organic chemicals in maternal nutrition and stress, and we had 4 race sex groups of pregnant women, and were there sex differences, and do maternal diet and weight gain mediate observed associations with chemicals? So the idea was to look at how the in utero environment is related to the growth of the fetus over time and then the outcomes at birth. So 34 women enrolled, 92% completed the protocol. 98% completed the study and had chart abstraction for delivery. Very high compliance. And again, MUSC was one of the sites, right, for this, so the 10 centers. So it's a fantastic cohort. This is the publication of the growth findings, and it's a very healthy group of women that were selected. There were really select inclusion criteria. And this is the publication. And this is a cohort profile of the cohorts of both singletons and twins. There are, in addition to 2,400, there are 171 twin mother dyads. And what they found was that significant race and ethnic specific differences in fetal growth during early pregnancy. They looked at biparietal diameter, head circumference, humerus length, and femur length. And there were differences for each race sex group as early as 10 to 16 weeks gestation, and they continue to diverge through gestation. What they found was that the race-sex differences primarily reflect the maternal intrinsic characteristics such as age, height, and subcutaneous fat, probably shaped by evolutionary processes. So up to this point, all of the growth charts for fetuses have been based on white kids, right? And what this says is that every single race-sex group, so 25% of the population is white, 25% Asian, 25% Hispanic, Latino, 25% black. And each one had a different trajectory. And so what this study really shows is that you just can't use white babies as a standard. But what else does that mean? How is the growth patterns and trajectories then related to the outcomes in the kids and these exposures? And so that's where we came along. We got together with Roger Newman and Kelly Hunt and I in public health and a whole group of other investigators, including investigators at each of the 10 centers, said, yeah, we're interested in following the kids. And so it's just having this many capable, brilliant researchers coming together is just fantastic. So this is Roger Newman and I don't know if many of you may know Dr. Newman. And so here's the hypothesis of what we propose for ECHO, is that there are environmental stressors, lifestyle conditions, and physiologic background. And so what we're really interested in is in the environmental epi-paradigm is look at air pollution, sediments, ingestion, and then look at vulnerability factors, and as well as all those things we measured during pregnancy, and see how those are related to an outcome. And so one of the nice things about a cohort study is we can look at this kind of paradigm as how all of these things are related to the outcomes in the kids. And so the conceptual motto for our study was that environmental stressors, chemical and non-chemical. By the way, I may be taking out some slides to shorten it up, but we had ultrasounds at 5 or 6 times during pregnancy. And it also measures even the organ size of the baby at every single stage, those 5 or 6 times, as well as maternal stress measures, diet, and we drew the blood of the mother 5 times during pregnancy. And at the baseline blood, we've measured a whole list of environmental chemicals, some of the ones that Demetri mentioned. All the newly emerging kind of persistent organic pollutants have been measured in the blood of the mothers. So we can look at those exposures during pregnancy and then see how those may be related to obesity and development impairment. And we can look at changes in fetal development and also how that may be related to risk, as well as looking at these environmental stressors. And we can look at race and ethnicity and gender and BMI, et cetera. So it's a fantastic study in terms of its capability. And so what we're really interested in is looking at, again, these child health outcomes at the end, but we can look at behaviors as well as environmental exposures. And we'll have the kids are now 4 to 8 years old when we bring them in. And so we'll be able to look at their outcomes here and then also ask them about their history of kind of any sort of disorders prior to that from the time of birth, look at behaviors and multiple mechanisms. So we'll be able to then follow these kids from this time over the next 5 years, and we can look at early life programming, postnatal shared environment, and how those are related to each of these outcomes in the kids. And so our 2 main outcomes that we're going to measure are, again, neurodevelopment and obesity and metabolic disorders in the kids. So again, it's a prospective study. So we have exposure, we've measured exposures, and the whole idea is to then follow the children over time for the outcome. And then these are, again, looking at maternal prenatal environment, exposure to persistent organic pollutants, stress, and nutrition, and look at child obesity and neurobehavior. And we also have a team of really innovative statistical approaches, modelers, to look at how these outcomes are related and to take into account all this multi-level exposure kind of in that model. So look at the fetal growth trajectories as well as environmental exposures and how those may be related to these outcomes in the kids. So hopefully we can identify kind of what the important critical points are that lead to this metabolic disorders in the kids, or for autism, for example. We may be, it's going to be hard for us because we only have 2,400 kids. This is the timeline in terms of, over the next 7 years. So we're now here at just finishing year 2. So we have the protocol up in place, and we already have 700 of the kids that we brought in over the in the 10 centers, and we'll continue that the rest of this year and starting in the next year to follow the kids. So unprecedented opportunity to understand the mechanisms of in utero exposures and their public health significance. So it's high quality data, the most detailed serial assessments of fetal development ever done, and phenomenal potential. We have blood samples and genetic information on the mother, and we'll also do this in the children as well. So compliments to the whole team, really, including Dr. Kelly Hunt, who's also a MPI, and Dr. Wapner, and the whole team of, just you can imagine 10 clinical centers, the teams that have to be in place to recruit, get the mothers and kids back in, and do these assessments. So the mission is to enhance the health of South Carolina's children's for generation to come, and cultivating a healthier tomorrow through leadership and research and prevention. And this is our new children's hospital. I think our goal should be that this sort of thing should be done. Every single mother child who enters this hospital should be given these kind of assessments, baseline, and follow, and basically set up a cohort of women and children to follow through time. And it should be done so that we can continue to learn from this and set up the best interventions for kids. And again, this is from ECHO, Enhance the Health of Children for Generations to Come. I love that logo. So with that, I'll end. I have lots more details on the study if people are interested, and list of publications that already have come out, et cetera. So with that, I'll end and open it to questions.